In the last video, we talked about our DNA-based lab technologies. Now, let's talk about the two RNA-based technologies. DNA is great, but it's unchanging. So your cells, all of the cells in your body have the same DNA. What makes kidney cells different from heart cells, different from brain cells, is the genes that they're expressing, the RNA that they're making through transcription. So measuring RNA will give you an idea of what the cell is like, what it's doing, what its active processes are at a given time. The issue is that RNA is usually very unstable. So our first step whenever we work with RNA is going to try to be to make that RNA more stable by making it double-stranded. We call this complementary DNA or cDNA. And to do this, we're going to take advantage of another viral enzyme, reverse transcriptase. And this one comes from retroviruses. Reverse transcriptase is going to work like polymerase, and it's going to make a complementary strand to the RNA molecule. So we're first going to make cDNA, and that's again stable. Now say we want to determine if a gene is being actively transcribed. Is there RNA for our favorite gene? Well, to do that, we can do a version of PCR that first has that reverse transcription step. So RT reverse transcription, PCR. And it's going to work the same as a normal PCR. We're going to first take our RNA, make cDNA, and then we will do PCR with primers to amplify whatever that gene is, and then we can run that on a gel and see if there's a band, same as we did with normal PCR. Now, RTPCR is great, but it's only going to tell you about that one gene. You can do a bunch of them, but it's going to take a while. Say you want to know about all of the genes being transcribed in a sample. This is when you would use something called RNA sequencing, or we like to be fancy here, we call it RNAseq. Everyone's doing RNAseq because everyone wants to know about all of the genes being transcribed in a certain sample. This creates a very large data set, just as a background for you guys, and usually requires special software to analyze and some computer training, but it's exciting stuff. So you want to do RNA-seq, you're going to take all of the RNA from a sample, you're going to make cDNA, you're going to shatter it into fragments, and then you're going to send it off. And they're going to do that Sanger sequencing on all of your cDNA fragments. And what they'll do is they'll then put them together using a computer and determine where those different sequences come from, what genes they are based on a known database. And the number of counts is going to give you an expression level. So if they find a lot of the gene for actin, for example, which is a cytoskeleton protein, lots of actin gene in your sample, well, that's going to correlate to lots of actin transcription. You're making a lot of actin. Whether that's useful information to you, that's where the computational, we call it bioinformatics side, comes into it. Okay, so in this video, we talked about our RNA technologies, and next time we're going to end with the discussion of protein technologies.